Okay, thank you, and I'm glad to be here. And the title sounds a little complicated, but what I want to do is actually, one second. Okay, I just want to give you kind of a general overview about tags, triacylglycerol, what they are and how they're made, and especially how they're made in biomass crops. And okay, maybe many of you already know what a triacylglycerol is, but if not, it's a molecule that has a glycerol backbone, has 3 carbons, and to each of the carbons, a fatty acid is bound. And okay, so we have 3 fatty acids. Typically in plants, the fatty acids have 16 and 18 carbon atoms, and they can be saturated or have no double bonds, or they can be monounsaturated as shown here, or polyunsaturated as in the bottom fatty acid. And when we talk about vegetable oils like soybean oil, they contain about 99% triacylglycerol. So we often will use the term vegetable oil and triacylglycerol, or TAG, interchangeably. Okay, and what I want to impress upon you is that these molecules are really great feedstocks for renewable diesel and sustainable aviation fuel. You probably have heard of biodiesel. That's the making the tags into what we call fatty acid methyl esters. But the technology now is moving towards something called renewable diesel, where the fatty acids are broken up into smaller pieces. We use catalyst and high energy to remove double bonds and split the fatty acids into smaller pieces. And if you break them up into small enough pieces, they can be used for sustainable aviation fuel, which is basically kerosene. And why do we actually care about tags or CABBI? Okay, in terms of bioenergy, tags are really among the most energy-dense molecules that exist in nature. And you can see here that this is comparing the kilojoules per gram. Okay, so a vegetable oil has about 37 kilojoules per gram. If you look at sugar, sucrose, that's half of the energy in 1 gram of sucrose that you get from TAG, and it's almost the same as you find in petroleum diesel, and a little bit less than in biodiesel, which is derived from the vegetable oil. And now we drive a lot in our cars. We use ethanol as to add with the gasoline, and even ethanol has a much lower lower energy density than tags. And in addition, tags are inert, so they can be stored kind of, they're not they have no, they don't affect the osmotic potential of the cell, so they can be stored in an inert form, typically as an oil body that's has a lipid monolayer and proteins that are associated with the oil body, but the vast portion of the oil body is tags. And so plants can sequester the oil in an inert form, and as you know, people and animals can also do this. And so this is why one of the reasons why we store oil, we store fat, because it's energy dense. We can store excess carbon away and use it when we need it. And we should look upon TAG and CABBI as a co-product. So we're making biomass, but we can readily extract TAG from the biomass, maybe using hexane or pressing it from the biomass. And so what's left of the biomass after the TAG is extracted can also be used for other products. So we look at the biomass as part of a biorefinery, where TAG is just one product that we can get from the biomass. And I think TAG is one of the things that separates CABBI from all of the other BRCs. I think we're the only one that's working on tags. Okay, so why do we care about tags beyond their energy density? Well, okay, so there's this huge demand right now for renewable diesel, and especially for sustainable aviation fuel. Okay, so the industry is moving toward production of renewable diesel. There's a lot of capacity there, but even more striking is the Sustainable Aviation Fuel Grand Challenge. Currently, in 2020, there was about 4.5 million gallons of sustainable aviation fuel produced from things like vegetable oils and animal fats. And we're looking to 2030 to go up to 3 billion gallons of sustainable aviation fuel produced per year. And by 2050, we want to be at 35 billion gallons of sustainable aviation fuel that's produced from sustainable sources like vegetable oils. And as I'll show on the next slide, that's a huge target to reach. But the technology to make renewable diesel, to make sustainable aviation fuel is already here. It's all worked out. The problem is that we need viable feedstocks. Okay, so I recently learned from talking to a grain processor that, okay, this goal of reaching 35 billion gallons of sustainable aviation fuel per year would take maybe 10 times more soybean production than we have currently. Okay, and so currently we produce about 5.6 billion gallons of vegetable oil. Okay, so we have to go, 6 times that to reach this target by 2050. Soybeans produce about 66 gallons of vegetable oil per acre. If we in CABBI could produce 10% oil in sorghum, and maybe we assume that we could get about 7 tons of biomass dry weight per acre, that would equate to about 184 gallons of vegetable oil per acre, more than maybe 2.5 times that of soybean. In a perfect world, that's what we would get. So we need a lot more vegetable oil. Okay. And so we see in CABBI that the biomass crops are a particularly possible platform for producing more TAG for these uses like renewable diesel and sustainable aviation fuel. Okay, in nature, seeds are what accumulate oil in plants. Seeds, soybean seeds, 20% of the weight can be TAG. Canola, maybe 42% of the weight is TAG. This is our current source of vegetable oils. This is already well worked out. People are trying to increase the oil even more in these oil seed crops. But what the big frontier is that CABBI is trying to tap into is to produce vegetable oils in the leaves and the stems, particularly in the stems of the biomass crops. And this is a huge challenge metabolically. Okay, the reason is that, okay, so all cells in a plant can make TAG. They can all make vegetable oil. The problem with the leaves and the stems is that the oils don't accumulate. Okay, so maybe with pathogen, maybe an insect chews on a leaf, maybe the plant is damaged by freezing temperatures. You have fatty acids released from membranes. They can be used to make oil, but that oil is quickly broken down. Okay, and so this is what we look at. We look at the kind of the balance between how much oil we can synthesize and store and how much is broken down. And if the breakdown exceeds the ability to produce and store oil, then we don't accumulate oil. We can produce all of the oil we wanted to, but if we are shifted towards the breakdown of oil, we won't have the accumulation. And that's the challenge that we face. And so we look at this concept of push, pull, and protect. And I'll show you that on the next slide. But the idea is that we try to push photosynthetic carbon into pathways like glycolysis that generate the substrates for fatty acid biosynthesis. This is called the push. Okay, so we're pushing carbon into fatty acid biosynthesis. The popular way of doing that is to overexpress a transcription factor called wrinkle-1, which turns on the expression of the genes for many of the glycolytic enzymes in the chloroplast, the plastid of the plant cell, and also some of the genes for some of the enzymes in fatty acid biosynthesis. So this is a very effective way for pushing carbon into the production of fatty acids. But then downstream, we want to pull the fatty acids into a storage form, into TAG. And typically, okay, so that would be outside of the chloroplast. And so that would be pulling the fatty acid chains for their storage on the glycerol backbone. And the final step in that pathway is catalyzed by diacylglycerol acyltransferase to make the tag. And so this is pulling the fatty acid flux into the tag. And then to accumulate the tag, so the tag would end up in oil bodies. We want to protect it from breakdown. We overexpress proteins called oleosin that coat the oil body and sort of protect the oil body from lipases that can degrade the oil. We can also downregulate lipases like this SDP1 that is involved in the degradation of fatty acids and or the breakdown of fatty acids from the tag molecule. And we can even go downstream and try to block maybe some or reduce some of the activity of enzymes and beta oxidation that are part of the recovering energy from the fatty acids. And so we want to be at a point where we exceed the breakdown with the synthesis and storage, and we're protecting the breakdown so that we're shifted towards oil accumulation. That's basically what we're trying to do. Push, pull, protect, and get accumulation of oil. Okay, and this is just putting the concept of push, pull, protect into action. Okay, so we're trying to make higher oil sorghum. And so this is one of the genetic constructs that we use where we are overexpressing DGAT1, overexpressing wrinkle1, overexpressing an oleosin. We have some other genes in here, and I won't go into that today, but this is just showing that with this construct in the field in 2021, okay, if we look at wild type sorghum, if we look at the amount of tag in wild type sorghum leaves, it's almost nothing. It's like 0.1% of the total dry weight. But in the engineered lines at the right stage of harvest, we can get up to about 5.5% tag in the leaf. And even more striking is the fact that if we look at the total fatty acids in a leaf, so fatty acids are in the membrane, lipids, and elsewhere, not just in tags. But if in the wild type leaf, you can have 3.5% of the total fatty acids present, but in these engineered lines, maybe you have almost 3 times the total amount of fatty acid. And so maybe it's not all about tag for producing sustainable aviation fuel. Maybe it's how much total fatty acid that we have. That's something that remains to be seen. But anyway, so we are making this oily sorghum now. And so the idea is to work with folks like Vijay Singh and his group and Conversion to figure out ways to more quantitatively extract the oil from the sorghum and use that for the bioprocessing and have the biomass left over for other potential uses. We're working with sustainability groups like Angela Kent and Wendy Yang to see if oil that accumulates maybe in the roots of the plant, maybe that can affect the microbiome of the soil. Because organisms like fat. It's not just that we store fat, but other organisms like to eat fat because there's a lot of energy that can be derived from it. And finally, we're looking at the future of oily sorghum. So our goal would be maybe to get up to about 10% oil in the stem. That would be great. And then working with the Singh group to work out the conditions for the extraction of oil on a larger scale and do this in a sustainable production system, working in a biorefinery and extracting that oil and using it to make sustainable aviation fuel, to make renewable diesel, and also other bioproducts that come from vegetable oil. Okay, that's all I have.
There's a question in the room. The next key step, our viewers want to know.
What's the next key step? To get higher levels of the vegetable oils and the sorghum. We're working, with folks like Jörg Schwender to understand what are the bottlenecks now, what's limiting the accumulation. And so we're kind of in that design, build, test, learn. We're in the learn cycle, trying to figure out what's kind of missing now to get higher levels of accumulation.